Welcome to the second video for the module on Chapter 17, where we're going to continue our discussion of cell replication, cell division. And so in the first video, we pretty much talked about DNA, discussed kind of its functionality, how it's structured, and how DNA gets transcribed and translated from the DNA molecule into messenger RNA and then into a protein. Okay. Now what we're going to do in the next couple of videos is go over the specific division processes, so mitosis and meiosis. So the processes we talked about before happen in the growth phase, happen in the synthesis phase, so replication, and these two processes, mitosis and meiosis, are specifically going to be that division phase. Okay. Now there are two types of cell reproduction processes that we have in our bodies. The first is mitosis, and remember we said that mitosis only happens in your somatic cells, so all of the cells in your body except for your reproductive cells, so our gametes or our eggs. I'm sorry, our gametes, our sperm or our eggs. And this process, this cell division process of mitosis, generates new diploid cells. Okay. Now, what we mean with diploid cells is that there are two copies of each chromosome. Okay, so remember we said that there are 46 chromosomes in the human cell, and so there are 23 individual chromosomes, and you have two copies of each of those chromosomes, so that makes the 46. Now, in mitosis, the cells start out with 46 chromosomes, and the two daughter cells end up with 46 chromosomes, right? So you have a cell, 46 chromosomes, it will divide, and you'll have two cells with 46 chromosomes, okay? This is termed as diploid, so having two copies of each chromosome is referred to as diploid. So diploid cells are 2N cells, okay? So they have two sets of chromosomes. Those two sets, one comes from the mother and one comes from the father. So you have a paternal and a maternal version of that chromosome. So for example, if you have chromosome number 1, right, that chromosome, so for that chromosome, you'll have two copies. One copy will be from the mom, and then another copy will be from the dad. Okay, so you'll have your paternal and then your maternal copy. Okay, and that's the case for the 23 chromosomes. So right, this would go numbers 1 all the way to 23, and you'll have two copies for a diploid individual. Okay, now for meiosis, this cellular division process generates haploid gametes. And so remember, this is only in the gametic cells. So for example, sperm and eggs. And those cells are haploid. So it goes from a diploid parent cell to haploid daughter cells. So they only have 23 chromosomes. So it goes from 46 to 23. Okay. And so in these cells, you're either going to have a paternal copy or a maternal copy. You'll have chromosomes numbered 1 to 23, but instead of having the two copies, maybe number 1 has the paternal copy and number 23, maybe that one is the maternal copy. Okay, and so it's either or. You don't have two copies of each chromosome in the gametes, and that's because when the gametes fuse, right, so when the sperm fertilizes an egg, the chromosomes will go into the zygote cell that's formed, the embryo, and so 23 plus 23, right, gives you 46. Now, if those gametes had 46 chromosomes each, if they were diploid, then the zygote would have 92 chromosomes, which would be very, very different. And it would cause, really big differences in how our chromosomes would be, potentially lead to, new species with different chromosome numbers, all of those things, right? And so in order to keep the chromosomes the same, the gametes, the reproductive cells, are haploid. And so haploid is referred to as 1N, okay? So diploid is 2N, haploid is 1N. And so what we're going to do with this video is we're going to focus on mitosis. Okay? So we're going to talk about that process. So as we mentioned, mitosis makes daughter cells that are diploid. They're also identical to the parent cell. So if you remember, in the S phase, during the cell cycle, the DNA is replicated, right? So if you have, again, chromosome number 1, and you have your paternal and then your maternal copies, okay? So 2 chromosomes, one from the dad, one from the mom, for chromosome number 1. Now during the S phase, they copy themselves. Right? And so we'll make another copy here. So let me actually erase this. Okay, so basically, that's terrible drawing. Hold on. I'll get it, I promise. Let's just do it like this. Okay, so now you have 2 copies of that chromosome, right? So you have one on the left, one on the right. And then the same thing would happen for the paternal copy, right? So now you have 2 copies of chromosome 1 from the paternal and 2 copies of chromosome 1 from the maternal. Okay, so that DNA is replicated. Now during mitosis, these replicated DNA segments, these replicated chromosomes, which are now called sister chromatids once they're replicated, will be split and separated, and you'll end up with 2 daughter cells that have the same DNA, so they'll be genetically identical to one another. Now mitosis has 4 phases that it has to go through during the cellular division process. And so after G2 phase, okay, you enter what's known as interphase, and then you actually go into prophase. And then after prophase, you go through metaphase, anaphase, telophase. And then a lot of the times you'll actually see telophase termed telophase / cytokinesis. Okay. And so the nuclear division, dividing that DNA, is the mitosis process. Actually dividing the cell itself, the cytoplasmic division, is cytokinesis. And that happens towards the end of that cell division cycle right after telophase. And so what we're going to do is we're going to talk through what happens during each of these different phases. So when we go into the cell division phase for mitosis, right, we first enter prophase. And let's say we draw a cell here. And in prophase, the nuclear envelope starts to break down. We're going to show that as kind of this dotted circle. Now, in the nucleus, you have your chromosomes. And now, like we said earlier, chromosomes have already been through S phase, so they're already replicated. Okay, so we're going to make this simple, and we're going to say, okay, we have one chromosome, like one pair, right? So number 1, basically, we have a paternal and a maternal copy. Okay, now in prophase, like I mentioned, the nuclear envelope starts to break down. What also happens is the centrioles start to migrate towards the poles of the cell. And they also start to develop what's known as spindle fibers. Okay, these spindle fibers will eventually grow out and attach to the sister chromatin. And that chromatin has condensed into visible chromosomes now. Okay. Now, after this happens, so after prophase, you get metaphase. And so with metaphase, right, you have your centromeres at the poles, and I'm drawing this dotted line here to represent the equator of the cell. So in metaphase, the sister chromatids will line up along the metaphase plate, where one copy of the sister chromatid is on one side of the cell and the other is on the other side of the cell. Okay, so basically like this. And then what happens is those spindle fibers will grow out and attach to the centromere of those sister chromatids. And then they will start retracting backwards. Okay, like that. And so once this has happened, the cell will then go into anaphase. Okay? And so anaphase is the actual separation of those sister chromatids. Okay? And so we'll have these centromeres. Okay. And then, so what happens is those sister chromatids will be separated and pulled apart from one another due to the shortening of these spindle fibers. Okay. So again, they're pulling them this way towards the poles. Okay. And as those, and then eventually they will be at each end of the pole, and you'll get the beginning of telophase, where, let me actually change this color. Right, where in telophase, it's basically the reverse of prophase. You have the nuclear envelope starting to form around the chromosomes. Right, so a red, right, went to each pole, so there would be a red in each one, and then a blue also went to each pole, so there's a blue in each one. Okay, and so the nuclear envelope will begin to form back. The mitotic spindles, right, so these structures will disintegrate, and then the chromosomes will uncoil, they'll uncondense, and then they'll revert back to the chromatin. Okay, and so after that, after telophase happens, then you get cytokinesis. Okay, so basically the same structure here, so the same cell structure in telophase, right? But now let's say the nucleus is formed, and we've got our chromosomes, right, in each one. And then once the nucleus has divided, the cytoplasm will split. And so basically you'll get this contractile ring to form at the midsection of the cell. And you can think about like putting a rubber band, a really strong rubber band over a balloon. And if you did that, it would indent, right, and pull that balloon towards the middle. And so that ring will continue to contract and get smaller and smaller. And you end up with a cleavage furrow in this area. And it basically, over time, that contractile ring will pinch those two cells apart. And so what you end up with, right, is or are two daughter cells with identical chromosomes. Right? So one copy, one chromosome with a paternal and a maternal copy. Right? So if you remember, that's what we started out with here, basically. Right? So before this, it would have been a paternal and then the maternal, and then this will undergo S phase, right, to replicate to get your replicated DNA. And so now we went from this parent cell to two daughter cells that are genetically identical to that parent cell. Okay. And so here's a figure that kind of shows that happening. Figure form and then actual micrographs of cells that are undergoing these processes. So this is showing two chromosomes. So this, you can just think of a big chromosome, which are these two. So you have a paternal and a maternal, and then a small chromosome, which are these two. And so you have a paternal and a maternal. So prophase, nuclear envelope breaks down. Centrioles line up at the poles, mitotic spindles form. Then in metaphase, they line up along that equator, the metaphase plate. The mitotic spindles, the spindle fibers, will go out and grab the centromere of the sister chromatids. In anaphase, those mitotic spindles shorten, pulling them towards the pole, separating the sister chromatids. And then now they're the daughter chromosomes. And then you'll have telophase, where the nuclear membranes will reform. And then cytokinesis is where that contractile ring is constricting to kind of pinch apart that cell into two daughter cells. And then notice you have big, small, paternal, maternal for each in each cell. So they're genetically identical to the beginning parent cell. Okay. So here is a video, a little animation, kind of going over this information.
This interphase cell is ready to enter the M phase of the cell cycle. Chromosomes have already been duplicated, and each consists of two identical sister chromatids. Each chromatid is one of the newly replicated DNA double helices, plus its associated proteins. In an interphase cell, there exists, just outside the nucleus, the centrosome, a cellular structure that acts as an organizing center for the assembly of microtubules. Mitosis begins with the events of prophase. In the cytoplasm, prophase is marked by the formation of the mitotic spindle, assemblies of microtubules emanating from each centrosome. The chromosomes condense into compact structures that can be seen with a light microscope. Before the cell enters metaphase, the nuclear envelope breaks down. After the nuclear envelope has disintegrated, spindle fibers from each pole of the mitotic spindle attach to one of the two sister chromatids of each chromosome. A tug-of-war is occurring, with spindle fibers pulling each chromosome in opposite directions. In the next stage of mitosis, metaphase, the chromosomes complete their journey to the middle of the cell. Each chromatid now faces the pole opposite that of its sister chromatid. At the start of anaphase, the link joining each pair of sister chromatids is broken. The spindle fibers shorten, pulling the chromosomes to opposite poles. This segregation of sister chromatids to opposite poles ensures that each daughter cell receives the same complement of chromosomes. In telophase, the final stage of mitosis, the mitotic spindle disintegrates and a nuclear envelope starts to form around each set of chromosomes as they return to their original uncondensed state. Once two independent nuclei have formed, mitosis is complete. Meanwhile, a cleavage furrow has begun to form, and immediately after the end of mitosis, the cytoplasm of the cell divides in a process called cytokinesis, forming two daughter cells.
All right, so that's a really good summary animation to kind of go over a couple times. If you have any issues or concerns with anything we've went over in mitosis. And one thing I would recommend in terms of studying for this is to draw the stuff out. Kind of do like I did in these kind of quick and simple drawings, just to see if you can draw it from your memory. And so if you can do that, you should be pretty good in terms of information that you need to know in mitosis. And you can also redraw this figure basically, and make sure you can label each part. You know, like what each thing is. So for example, sister chromatids versus, the centromere, the centrioles, the mitotic spindles, the daughter chromosomes, when they're a chromosome versus a oh my gosh, I'm blanking. Chromosomes versus I'll think of it in a minute. The uncondensed chromosomes. I cannot think of that word right now. And so make sure you kind of chromatin. There you go. Make sure you know the differences between those. Okay. Brain fart. I'm so sorry. And so with this cellular division process, mitosis, kind of are gone over. If you can understand mitosis, then that will help you out a lot in terms of understanding meiosis, because meiosis is a cellular division phase that happens in two different division cycles or two different division processes. The first division phase is really different from mitosis, but the second division phase is relatively similar. So if you can understand and get a grasp if you can get a grasp of mitosis, then that will help out a lot with meiosis. Okay, and so with that, we'll end this video here, and then we'll pick up in the next video going over the second cell division process, which is meiosis. So we'll see you in the next video.